What is prokaryotic cell? Prokaryotic cells are simple, unicellular organisms that lack a defined nucleus and membrane-bound organelles. They are found in organisms such as bacteria and archaea. Despite their simplicity, prokaryotic cells perform all the essential functions of life, and understanding their structure and components provides insight into how these organisms survive and thrive in diverse environments. 1. Cell wall. The cell wall is a rigid outer structure that provides shape and protection to the prokaryotic cell. It is primarily composed of peptidoglycan, a polymer made of sugars and amino acids. This composition varies between different types of prokaryotes, leading to differences in their structure and staining characteristics. For example, gram-positive bacteria have thick peptidoglycan layers, whereas gram-negative bacteria have a thinner peptidoglycan layer and an additional outer membrane. The primary function of the cell wall is to maintain the cell's shape, protect it from mechanical stress, and prevent osmotic lysis, ensuring the cell doesn't burst in hypotonic environments. It also serves as a barrier to harmful substances, including antibiotics. 2. Plasma membrane. The plasma membrane, also known as the cytoplasmic membrane, is a selective barrier that regulates the entry and exit of materials into the cell. It consists of a phospholipid bilayer with embedded proteins. The hydrophobic core of the membrane prevents the free passage of ions and polar molecules, while the embedded proteins facilitate the transport of substances across the membrane, either passively or through active transport mechanisms. In addition to regulating transport, the plasma membrane plays a key role in cell communication and maintaining the internal environment of the cell. It also anchors enzymes involved in energy production, allowing for processes like cellular respiration and photosynthesis in some prokaryotes. 3. Cytoplasm. The cytoplasm is the jelly-like substance inside the cell that contains all of its internal structures. It is composed mainly of water, salts, proteins, and nucleic acids. The cytoplasm is where the cell's metabolic processes take place, including glycolysis, protein synthesis, and various enzymatic reactions. In prokaryotic cells, the cytoplasm supports the ribosomes and serves as the medium for the cell's chemical reactions. It also helps in maintaining the shape and consistency of the cell by providing a platform for the proper positioning of other cellular structures. 4. Nucleoid. The nucleoid is the region in prokaryotic cells where the genetic material, or DNA, is located. Unlike eukaryotes, prokaryotes do not have a membrane-bound nucleus. Instead, the DNA in the nucleoid is typically a single, circular chromosome that carries all the genetic instructions necessary for cell function and reproduction. The nucleoid region lacks the complex organization seen in eukaryotic nuclei. In prokaryotes, there is no nuclear membrane, and the DNA is directly exposed to the cytoplasm. This simpler organization allows for faster replication and genetic exchange, which is vital for survival in changing environments. 5. Ribosomes. Ribosomes are the molecular machines responsible for protein synthesis. In prokaryotic cells, ribosomes are smaller than those in eukaryotic cells, typically referred to as 70S ribosomes, compared to 80S in eukaryotes. They are made of ribosomal RNA, rRNA, and proteins, and they exist freely in the cytoplasm. Ribosomes read messenger RNA, mRNA, to assemble amino acids into proteins through a process called translation. These proteins play crucial roles in the cell's metabolism, structure, and function. The efficiency of protein production in prokaryotes allows them to rapidly respond to environmental changes. 6. Flagella and pili. Flagella are long, whip-like structures that extend from the surface of some prokaryotic cells. They allow for movement, enabling the cell to swim toward or away from certain stimuli, a behavior known as taxis. Prokaryotic flagella are made of a protein called flagellin and are powered by a proton gradient across the plasma membrane. Pili, or fimbriae, are shorter, hair-like structures that extend from the surface of the cell. They are involved in attachment to surfaces or other cells, and some pili are used for conjugation, a form of genetic exchange between prokaryotes. Pili can also play a role in biofilm formation, allowing prokaryotes to adhere to surfaces and form communities. 7. Plasmids. Plasmids are small, circular pieces of DNA that are separate from the chromosomal DNA. They can carry genes that provide advantageous traits to the bacteria, such as antibiotic resistance, virulence factors, or metabolic capabilities. These extra chromosomal DNA molecules are transferred between bacteria through processes like conjugation, promoting genetic variation. Plasmids are important in the context of antibiotic resistance, as they can spread resistance genes rapidly among bacterial populations, contributing to the challenge of treating bacterial infections. 8. Inclusion bodies. Inclusion bodies are aggregates of various substances stored within the cytoplasm of prokaryotic cells. These substances can include nutrients, minerals, and even waste products. For example, bacteria may store glycogen or lipids in inclusion bodies, which can be used later when external nutrient sources are scarce. In some bacteria, inclusion bodies contain velutin, phosphate reserves, or sulfur granules used for energy storage. These stored reserves are vital for prokaryote survival in fluctuating environments. 9. Endospores. Certain prokaryotic cells, especially gram-positive bacteria, can form endospores under stressful conditions, such as nutrient depletion or extreme temperatures. An endospore is a highly resistant, dormant structure that protects the bacterial DNA from harsh environments, allowing the bacterium to survive for long periods without growing or reproducing. When environmental conditions become favorable again, the endospore can germinate and return to its active form. This ability is critical for survival in extreme environments like hot springs or during antibiotic treatment. Examples and significance in nature. Examples of prokaryotic organisms include Escherichia coli, E. coli, Streptococcus, Salmonella, and archaea like thermophiles and halophils. Prokaryotes play a fundamental role in many ecosystems. They are involved in nutrient cycling, such as nitrogen fixation, which makes nitrogen available to plants. They also contribute to the breakdown of organic matter, and some are even used in industrial applications like fermentation. Prokaryotes have immense ecological and evolutionary significance, shaping the environment and participating in symbiotic relationships with plants and animals. Conclusion. In summary, prokaryotic cells, while simpler in structure than eukaryotic cells, have all the essential components necessary for life. Their ability to rapidly adapt and thrive in a wide range of environments makes them one of the most successful forms of life on Earth. Understanding their structure and functions provides insight into both their unique biology and their pivotal role in nature. If this video is helpful, don't forget to like, subscribe and share.